For some people, it's difficult to imagine that the sea surface macro layer, this SML, exists.
Do you think it's the most important millimeter on the planet?
I think so. It's like the skin of the ocean. It's key because if you want to pass from the ocean to the atmosphere or the other way around, you need to go through this skin.
Gas and heat and energy transfers have to go through it. There's no way to go around it.
We kind of can't see it, but you can sample it. And then we can gather data out of what's happening in this very, very thin layer on top of the water.
It's not static, it's always interacting with the atmosphere, but also with the underlying bulk water. As a biochemical reactor, producing unique organic molecules, recycling organic matter, being the basis of a unique microbial community.
Depending on how thick it is, depending on what's in there, depending on which organisms thrive in there and stuff, flux of energy, like the waves, how strong the waves are, the flux of gases, the flux of CO2.
We have a lot of models about climate change, but in this model we do not have the small-scale convection processes. There's a big knowledge gap about it.
Normally the oceanography community has been studying the surface ocean, measuring at 2 or 3 meters depth, right? Which is like a dermatologist trying to study the skin doing an X-ray, right? They don't know anything about the skin because you are too deep on your body. Fluxes from the atmosphere to the ocean has been forever calculated from this 2, 3, 5 meters depth. And this is what we want to improve.
The BAS group wants to study this SML, a slime-like matrix full of algae and microbes, a layer that varies from 0.1 to 1 millimeter thick, creating slicks which shape wave properties and gas transfers and cover much of the world's oceans. But how can you go about studying an intensely dynamic habitat which is changing all the time?
We have this MESOCOSM facility here to conduct experiments in controlled conditions.
A MESOCOSM experiment is when you want to recreate a little piece from nature in some artificial facilities. Like this pool where we are trying to recreate a natural bloom to see how the surface macro layer is changing through time.
Meso means something in between. So we have kind of natural conditions because we used water out of the Jade Bay. But on the other hand, we are not out in the Jade Bay, so we don't have the influence of water flux, we don't have the influence of too much wind, rain.
The idea of the MESOCOSM experiments is to facilitate this collection of data.
We can do a time series, so we can study the same water mass for, in our case, 30 days. We can integrate a number of sensors without, the rolling and the pitches of a research vessel, observe all the processes happening right at the surface.
It's almost like working on a ship. It has the same spirit. Actually, it's also my first kind of experiment in that way. So it's a nice experience.
You have a few different sensors that measure temperature, salinity, turbidity, chlorophyll, oxygen, for example. And the water is then released back to the tank. So you have like a flow-through system.
Also, normally oceanography instruments are big, so you cannot measure the first 1 millimeter of the sea surface. Here in this experiment, we use microprofilers. Microprofilers has a tip size of 200 micrometer, so you can profile the first 5 to 7 millimeter of the sea surface.
So this kind of approaches or observation will be very hard if we try to do it in the open ocean because of the winds, the waves.
It's a round-the-clock effort to better understand the properties of the SML. Which aquatic plants like algae are in there? Who are the bacterial communities living there? And how are they changing? How does the SML affect gas transfers from the water to the atmosphere and back? And how are the physical structures of the layer changing with different conditions over time?
The way we are constructing this story is with daily samplings. Taking these samples very early in the morning before the sunrise in order to see the signal from the night, and also we take samples at the afternoon after the facility has received a lot of sunlight, seeing the signal day and night.
This is a metaphor I use for sampling actually, because people ask me why you dip a glass plate under the water and then you just lift it. Why you assume there's the seesaw-of-Smykley attached to it? I'm like, it's like a tablecloth, you just lift it and it attaches to the surface of my glass plate. It's hydrophilic, which means it attracts water, while the seesaw-of-Smykley is kind of like willing to attach to my glass plate.
Every day you have your sampling times and everybody's there with their bottles. We get like a little bit of SML and a big chunk of bulk, and we go back to the lab.
In the beginning of the experiments, we filtered this 13,000 liters of water for 3-4 days to have a really clean water mass.
The tank was completely clear, you could see the bottom.
After that, we actually added nutrients to trigger the remaining phytoplankton to grow.
What we have here is a series of samples, and what we are seeing is from the very beginning just filtrated water. And at this point in time, we decide to add some nutrients into the MESOCOSM experiment. And then a few days later, we start to see a lot of things growing up. We are seeing here the phytoplankton bloom. And this phytoplankton bloom is maintaining through time.
You can really see processes which you may not see. In the sea, we cannot add nutrients to trigger a bloom, for example. We just have to go and see, and to be lucky sometimes to find the right conditions.
Everyone gets their sample of the SML and are studying it from a range of perspectives.
When we get a sample from the surface microlayer, what we are here doing is an automated microscopic analysis. At the beginning, you saw only these small needle-like diatoms. There was 1, 2, 3 or 4 per sample, I would say. In a couple of days after we added nutrients, they exploded in numbers. Here we have, in the mesocosm during the bloom, 1 million cells per milliliter, and that's quite striking, I think. Almost all particles are phytoplankton cells. It's always striking that these small algae are occurring in such high numbers during blooms. This shows actually that you have almost half of the plants which are producing half of the oxygen on the planet. They are not visible by the naked eye, actually, and that's always amazing, so that these small plants are so important.
The SML, I didn't know such a thing existed, and I read a lot of literature about it. I was like, okay, maybe, but now I can actually see the difference. So this is the surface, the SML, and you can see there's quite a lot of particles in here. I don't know. Most of these are algae cells, and this is the underlying water. It looks like regular seawater, so it doesn't have as much of a slimy texture to it. The SML, it's a very, very specific condition in that habitat, and you have these communities which stick to that and which you don't really find below. I just read that until now, but now I'm seeing it. So yeah, we're all pretty excited, and yeah, believe in SML. It exists and it changes a lot of things in the ocean for sure.
Most of the PhD students from the project are here, and we are working on almost the same topic, but like with all these different parameters and all these different points of view.
I think it's a lot of work right now. So right now I would say it's my first home, not my second home. I think for me the interesting part is how we can see the change over the whole weeks. Here in SURF I'm working with infrared cameras from the top to the surface and look to the horizontal structures of the water body, because we can see it really clear that we have something like eddies or something like that on the surface, a different structure than at the beginning and at the end. And I think that's what I hope to see in the end. And yeah, let's see.
So we look on optical properties of the material in the water, which is basically how much of light is absorbed and also how much light is scattered.
Yeah, so this is the machine. It has a laser. You can see the green light
shining through the water. The light comes from the right side, and on the left side there's a detector that measures the scattering. You see now that the laser beam gets brighter because there are now particles in the water that actually scatters more to the side. And that tells us, first of all, how much of the phytoplankton is in the water, and also whether there's a specific phytoplankton community that actually lives in the microlayer, which is actually now happening in the experiments.
And for me, what is interesting is the bacterial community living in association with these algal cells. So we want to see who they're associating with and what kind of community is growing and how it is changing over time as it is being exposed to more nutrients, more sunlight, UV, all of these changes. So I take the seawater, trap the cells and the DNA on these membrane filters, extract the DNA, and then look at the community, whether it changes at all, or do we find some very strange bacteria.
Some people say the communities are absolutely unique there. The other people say, no, it's not really that unique. Thanks to the new sampling techniques that we're applying here, I will have enough volume to apply some methods which were not possible before, such as metagenomics and metaproteomics. And that way, maybe we'll finally figure out what makes the SML such a unique environment and if it's really that unique for the bacteria that inhabit it.
Would you make a bet whether it will be unique or not?
I'm not a gambling man, but my bet would be that it is unique.
When many people see the ocean, they just see water, right? So, but when you understand all of these processes occurring all the time, day and night, you can recreate what is happening in there. Like you are seeing a very calm sea, but in your mind you are seeing exchange of gases. When you understand this, you open a window to the world.
Looking into CO2 only is a very simplified approach. In science, we try to look at the full system of things, and then CO2 is just too simple. There's many more gases that have an effect, and especially for trace gases, there's this thought that we really underestimate the effect of the SML. In terms of trace gases, I would think it's unique because it's so very hard to sample. So basically we don't know anything about it.
You will have some places where there's a lot of phytoplankton and there's other places where it's lacking. One part of the world, the ocean is absorbing the gases from the atmosphere, and other places can act like a source of these gases. That depends on the amount of phytoplankton or biological processes that are occurring inside the surface microlayer.
Then if we don't take into account the SML, we might actually under or overestimate the effect.
So I think what we want to do is actually provide basic knowledge so people could include it into models.
Well, SURF has the advantage that you can control things much better than here in the field, where you have this constant mixing and exchange processes with the atmosphere.
Every day the team goes out on a boat alongside an autonomous sampling catamaran.
Well, I built my first model boat with my dad as I was 7. Am I qualified? We have our daily sampling here in the Jade Bay. We come out every day as long as the weather conditions allow us. We just go always to the same spot, always at high tide, and then we will see what we find. Slicks, which are areas where capillary waves are damped and very rich in organic matter. Sometimes we have rain, so we just take what we get. So we have a set of rotating glass discs immersed in the water, rotating through the surface, picking up the sea surface microlayer. We are collecting it in a vessel and pump it through our sensors.
That is my favorite part. It feels really cool to get the samples from the field, and then we are doing sampling in our MESOCOSM facility on daily basis, and we can be sure like, okay, these processes are definitely happening in the natural environmental conditions also.
And later we will do this glass plate sampling to get a comparison between the manual technique and the catamaran technique. The name Halobates, it's a marine water skater. From the only insect in our world among 700 million which lives the whole life on the open ocean. And since we are sampling the sea surface, we thought that's the best name.
So the approach that we are doing here is kind of limited. We are not considering the wind influence or the waves. You need to think in the physics, in the dynamic of the ocean.
And in a few months, we will go to the wind wave tunnel facility of the University of Hamburg to look at the interaction of the microlayer with the overlying air and the underlying bulk water. So these two experiments complement each other.
And then in 2024, in the summer, we will also have a big cruise, we call it, to really run all these methods that we're testing now.
By comparing all these measurements and experiments, this is the great value of the design of the project.
Next year on our big field campaign, we want to use this knowledge to explain what we observe in the field.
That way we can make that all-encompassing study of the seawater microlayer that everyone is so excited about.
The fun part of studying the surface microlayer is that you have the opportunity to explore the ocean. You need to hunt these spots, slicks, where when the surface microlayer is very developed, it's like when you want to see the behavior of elephants, no? In this case, we are trying to see the behavior of the surface microlayer, how it's growing, taking the carbon from the atmosphere, how it's releasing it back again to the atmosphere.
The sea surface is really important because it covers 70% of the Earth's planet.
And in this way, you can start to recreate a story of the whole world.
There's two types of SML. There's a visible SML and invisible SML. The invisible SML you don't see. You just believe science, crazy science like us telling you there's a SML there. But the visible SML is it's a phenomena that it's so much organic matter that you don't have these normal waves, these very small waves that is normally on the ocean. So it's dampening this. And this is when you see the sea like a mirror. This is how fishermen describe as leaks. Today we have the sea like a mirror. And the sea surface macrolayer, you don't know the name, right? Like everyone knows the name of a wave. No one knows the name of a slick. But once you know, you see it and you say, oh, I see it my whole life. And actually they are beautiful, right? Very beautiful.
Well, so far everything went so well, but I know that also a lot of students are really looking forward to next week because it was really hard work, 24/7 basically. I just look forward to the end of the next week because then we can jump right into the data for the new understanding of the microlayer and in interaction with the atmosphere.